Hello, College Biology. This is protein part 2, and we're going to talk about the polymer and how it becomes a protein. In the previous video, we talked about the amino acid monomers. Those monomers will be linked together with peptide bonds that the ribosome helps make that happen. Well, if you take lots of peptide bonds between lots of amino acids, you have many peptide bonds, and that amino acid chain is called a polypeptide, and that is our polymer for proteins. And the polypeptide is not the end of the story. You don't just make a polypeptide and there you have protein. That polypeptide has to fold into specific shapes. The jobs that proteins have depend on their shapes. So that polypeptide has to fold itself into a specific shape for the cell to have the protein that it needs. So there are 4 stages of this folding process, and the first is called the primary structure of the protein. And that is simply defined by the order of the amino acids in that polypeptide. In this particular example, we have 129 polypeptides, and they are all listed here, and these are abbreviations for all of their names. We know that there are 20 different types, so they can be shuffled and reused multiple times. This order matters because some of those amino acids are polar and some of them are nonpolar. And they're going to have interactions with each other based on that, which will lead to a specific shape in the folding, which will allow the protein to have a specific function. So the primary structure is the order of the amino acids. Now, the order of the amino acids will influence the secondary structure. Secondary structures are very specific shapes that form due to polar amino acids forming hydrogen bonds with each other and possibly nonpolar amino acids having hydrophobic interactions with each other. And 2 shapes that continually pop up in protein are the alpha helix and the beta sheet, sometimes called the pleated sheet. And what these shapes are, the alpha helix is a spiral shape that comes from polar bonding between R groups of the amino acids, hydrogen bonding, at regular intervals. So if you've got a polar amino acids at regular intervals, they'll be attracted to each other and it'll result in a spiral shape, and that's the alpha helix. The beta sheet is also caused primarily by hydrogen bonding, but as a consequence of how they are arranged, the polypeptide folds over on itself and forms hydrogen bonding between strands. It's much like cellulose strands do into the cell wall, only it's a flat structure. And so those shapes will form spontaneously due to the arrangement of the amino acids and their polarity. So that primary structure will lead to these specific secondary structures occurring. Tertiary structure is essentially everything else. Once the secondary structures are formed, you still have a polypeptide, and those polypeptides the rest of that polypeptide is made of amino acids, and they will also fold, and they have some more complex things going on. In addition to hydrogen bonds and hydrophobic interactions, there can be covalent bonds between sulfur molecules that show up, and there can be ionic bonds that show up. And so all of those contribute to the rest of the polypeptide folding around the secondary structures to give us this fully folded polypeptide. For some polypeptides, this is it. We are now fully functional. We are a protein, and we can do our job. Maybe we're a structure protein, maybe we're an enzyme, maybe we're a transport protein, I don't know, but we're done. But for most proteins, we have to go on to a 4th level, which is quaternary structure, which is simply saying 2 tertiary structure, 2 fully folded polypeptides are attracted together and unite, and that becomes the final protein structure. And now this is going to be able to do some job for the cell. So to quick review, primary structure is all the order of the amino acids. The secondary structure are specific shapes, the alpha helix and the beta sheet, which are a consequence of the hydrogen bonding of the R groups from the primary structure. The tertiary structure is what everything else in the polypeptide is doing, folding around the secondary structures. In quaternary structures, when you have 2 polypeptides that have fully folded, uniting together to become something useful. So for our notes, the polymers of protein are amino acid chains called polypeptides. Polypeptides must fold through 4 stages to become a functional protein. The primary structure is the order of the amino acids in a polypeptide. The secondary structure occurs when polar amino acids form hydrogen bonds and nonpolar amino acids have hydrophobic interactions resulting in specific shapes. And two of those shapes are the alpha helix, which is a spiral shape formed by having polar amino acids at regular intervals, and the beta sheet, where the polypeptide folds back on itself several times due to hydrogen bonds forming between polar amino acids. Tertiary structures when the rest of the polypeptide folds around the secondary structures due to their polarity, and for some polypeptides, this is the final functional protein shape. And finally, quaternary structure is when multiple tertiary structures are attracted together to form a functional protein. Each polypeptide in the final protein is called a subunit. Pardon the bell. I hope this helps. Struggle well.